Thank you. An excellent introduction. So for our second contribution today, we have Fabio Benedetti from ETH. So Fabio, I will just hand straight over to you to tell us what you want to tell us. Thank you.
Yes, thank you, Adrian. If you can hear me well and see the slides, please give me a thumbs up.
Yeah, you're fine.
Cool, thank you. So I'd like to apologize in advance because I won't be talking about mesopelagic or any deep sea stuff in this talk. And I'm also apologizing here for myself because this is actually the topic I wanted to study when I first got into biological oceanography. To begin with, I wanted to study deep sea animals, exactly like Henk-Jan just presented, but you know, life takes you places. And for myself, it turned out that life made me a shallow surface person. But that's fine. I hope I can dive back in there in the future. And yeah, I'm looking forward to contributing more to the science of the deep sea. Today I'll be currently, well, life took me in Zurich right now. I'm a postdoc in Nicholas Gruber's group at ETH Zurich, where I'm mainly supervising projects on the climate change impacts of surface plankton species richness and composition. In our group, we have various topics on the matter, but Aidan Martin kindly invited me to present one of the studies we recently published, and this is what I'll talk about, but please see this as an overview of all the studies we're ongoing on this project in our group. And I would first like to, of course, thank the main contributors to this study, which are Mike Vogt and Nicky Gruber, my main collaborators in the group, Urs, a PhD student in our group, Damiano, who's now a postdoc in Copenhagen, and the professor Niklas Zimmermann, also in Zurich. So, talking about climate change and plankton. So the study I'm going to talk to you about is embedded in climate change science. So of course, it's all about trying to understand how the system, that is the ocean, will respond as we emit more and more CO2, anthropogenic CO2 in the atmosphere. And a lot of stuff has been accomplished, and this is nicely summarized in the latest report by the IPCC, which I'm just citing and showing some pictures, some graphs from here, but you all know this better than me, that now, thanks to the immense effort that has been carried out by the community over the last decades, we're able to model how the system can respond to future increases in temperature. So overall, I'm just showing some main maps here according to so the simulations of net primary production on the top, total animal biomass in the middle, and maximum fisheries catch potential at the bottom, according to two different emission scenarios, one with mitigation, 2.6, and one that is more business as usual, a high emission representative concentration pathway. And most of the models go towards the same direction, which is that we expect a decrease in global biomass of marine animal communities, the production, the catching fisheries, and so on, so a loss in economical activities for the 21st century as a response of carbon emissions by the human societies. And these changes propagate from the basis of the food web, which are the plankton, which I will talk about more later, and across the food web, so to small fishes and higher trophic levels like seabirds, mammals, and larger fishes. And so as a response to climate change, we expect global scale biomass for our marine animals to decrease by almost 15% at the global scale. And this will likely transfer into a decrease in the maximum catch potential of fisheries by 20 to 24% by the end of the century under a high emission scenario. These results are obtained by a combination of very different models. And let's say that the plankton is there as a pivotal role because it forms the basis of the food web and all the organic matter that is then provided to the higher trophic levels and that will sink in into the deeper ocean. So what we're trying here in the group is try to better describe the diversity in the plankton and to maybe help improve those ecosystemic models that generally only summarize the variety of plankton diversity and plankton traits through a few boxes, a few PFTs. And we're trying to show how the system will change in terms of species diversity, which will translate in terms of species traits, and so loss or gains in ecosystem functions. So the various dimensions of plankton diversity, so I'm talking about dimensions in terms of variables that you can use to describe biodiversity in general. So commonly people use species richness, the number of species, but you can also have a look at this in terms of species compositions, trait compositions, stuff like size structure, the proportion of large species to small species, the proportion of calcifying species to silicifying species, and so on, but also species interaction. And for plankton, all of this is shaped by climate and ocean dynamics, which we know. But what we don't know that much is how the drivers change across scale. So as you go from the mesoscale to the basin wine or the global scale, the drivers of species diversity or trait variability are likely to change because the processes behind are changing. And the way we can capture those processes are also changing. And all these changes in a way that remain poorly understood. Although it's important to study that because plankton diversity, of course, governs important processes from nutrient cycling or to the intensity, the efficacy of the biological carbon pump or the food provision to fishes. For instance, the system does not behave the same if you have a phytoplankton community that is dominated by large silicifying diatoms than when it is dominated by smaller calcifying coccolithophores. The same for zooplankton. The system, the food web doesn't function the same if you're dominated by small ambush feeding copepods compared to larger grazing and predatory krill in the Southern Ocean. And one of the caveats we have with the models that are currently being used by the IPCC is that these ecosystem models usually pull the result of diversity. Just to take an example, in our own group, the ecosystem model that is used by most of the PhD students often comprises only one box for zooplankton. So we have to do better about this. One of the approaches that has been going on for several decades is, so how can we better model plankton diversity to understand their contribution to ecosystem processes? One way that has been developed, especially by the people at MIT with the Darwin model, which I found very interesting, is about increasing the number of boxes, so the number of plankton functional types in the models, and to have some sort of community assembly model that is all driven by mechanistic processes. That is a mechanistic model, and that allows you to test and see what are the emerging properties of the system and what are the emergent patterns the patterns that emerge after running the model. And I'm just showing a few examples here from two relatively recent papers that show patterns of phytoplankton diversity. I won't go in depth into these studies, but I encourage people to have a look at them. Another approach that one can use to understand plankton diversity and the patterns in time and space is to model the diversity patterns directly using statistical models based on observations. Like I'm also here giving a few examples by two papers that emerged from the Tara Oceans cruises that you might have heard about, a paper published in 2019 in Cell and in 2020 in Global Ecology and Biography. And what these authors did is that they, thanks to the immense effort that has been put in the Tara Oceans expedition, they sampled at each station. There are approximately between 100 and 200 stations worldwide. They sampled the DNA in the seawater, and they performed high throughput sequencing to measure diversity directly. And from these, they could derive statistical models that try to explain diversity patterns in terms of parameters such as temperature or chlorophyll air concentrations. And from that, you can make projections in space and time. The problem with these approaches is that at very large scales, usually you only have one or two field cruises that cover such a wide range if you want to look at global pictures. So they're rare, and especially they're very hard to compare to one another because the sampling strategy usually completely differ. and they usually focus on certain groups and not on the full community. And they, of course, since there are cruises, single cruises, they only provide a snapshot of the system. And if you want to try to have a global picture that resolves seasonality and so on, you tend to have here a limited temporal scale and observations, which are also heavily influenced by local factors such as mesoscale or diurnal variation. And this is not something, and this is something that then is opposed to the fact that you're looking at global patterns. But also, again, encourage people to have a look at these papers. And so the alternative strategy we opted for in the group is that we're going to use statistical models based on observations, but we are going to collect from online public databases as many as occurrences as we can in a limited amount of time. Of course, I only have one life. And we hope to mine this dataset to model each individual species environmental envelope. So this is what is called species distribution modeling or environmental niche modeling, which was a work that was initiated years ago, especially by Grégory Beaugrand when it comes to copepods. So what I'm going to show here and that we're going to generate with these approaches are not maps of abundances or biomass, but rather maps of habitat suitability, which help us so quality of the abiotic habitat that help us estimate monthly community composition in space and time, and from which you can derive climate change projections, of course. I'm just very briefly showing here the data sets that we have at NAN and from which we perform the species distribution modeling. On top, I'll provide an overview of the sampling effort of the phytoplankton dataset, and at the bottom for the zooplankton dataset that I implemented during my first years at Zurich 3 years ago. And basically, from this dataset, we can retrieve occurrences, so information in space and time of where a certain species was present. And the data range from the latest 19th century to until today. As you can see directly from the map, there are huge gaps in sampling effort. Generally, you don't have data for the basins that are very far away from the coast, so typically tropical Atlantic and tropical Pacific. And we have more data in terms of quantity and coverage for zooplankton than for phytoplankton, as it is relatively easier to sample in zooplankton. For the phytoplankton, the data set at the end, the clean version retains almost 1 million of observations in first observations. That span in the end more than 600 species or taxa. And as you can see here from the percentage, the relative contribution of the groups, nearly half of them are diatoms and nearly half of them are dinoflagellate, and the rest it's basically haptophytes, so coccolithophores and Phaeocystis. So what you can see already here, there is a bias in the taxonomic coverage of the dataset, meaning that we're mainly looking at modeling microphytoplankton species, so rather large stuff. So we're not covering bacteria and nano and picophytoplankton. And again, so for the zooplankton, most of the data come from copepods, which are the main group in the mesozooplankton in terms of abundance, but also diversity, so it makes sense. But we also have substantial contributions from groups like etognat, large crustaceans, which basically are krill and shrimps, but also theropods and jellyfish and so on. And can I click? Yes. And both these data sets are available. The first was published in 2020 and is available on Pangea, and the other I made it available in Zenodo recently. So briefly, the species distribution modeling framework. So what do we do how can we have diversity estimates from species level maps? What we do here is usually first, we aggregate the biological data in the context of our study. Those were presences, not abundances or biomass, and we match them at a climatological scale to environmental predictors such as sea surface temperature, macronutrients concentration, mixture depth, and so on. And then we use various statistical algorithms of various types or various degrees of complexity to try to estimate their response curve. So how their habitat suitability changes with gradients of, as I said before, the predictors that are included in the model, such as temperature, irradiance, mixture, and so on. We perform various sets of random splitting. So usually we train the models on 80% of the data and validate them on 20% that are randomly split, and we do this several times. That serves as a basis for cross-evaluation. And then we project for each of these models and for each species maps of habitat suitability that can be in space and time at various scales. And for each map, we have one species. And so basically, if you stack all the species maps all together, at the end, you come with an estimate of an assemblage or a community composition. And as a second step, from these assemblages composition estimates, you can then derive diversity indices and changes in composition, space and time. And as a further test of validation, we check how these diversity patterns match with previous observations to see if they're consistent and so on. So the models are, of course, heavily influenced by the type of predictors you put as a predictor. So I'm just going to quickly explain how we choose those. This is a very state-of-the-art niche modeling procedure. So we first exclude all those predictors that present a pairwise correlation because we want to avoid highly collinear predictors, for instance, nitrate and silicate at the global scale, or surface temperature and surface oxygen to be included in the same model. Otherwise, we inflate some parameters and we bias the models towards certain areas. We define then to further narrow down the pool of predictors, to further narrow down the number of predictors included in the model because we want to avoid model overfitting, which is a very big issue in species distribution modeling. We have to narrow down the final 5 or final 6 sets of predictors that we want to include. For this, we run several generalized linear models and then random forests, which we'll all use for the projections to examine the relative importance of each predictor for the species, and then can examine the ranks. And after ranking those variables, we then make a first set of species distribution models. And at the species level, we go through every map and we make sure that they make sense. That Southern Ocean species are projected in the Southern Ocean and not in the Arctic, and so on and so on. And just for information, so in the maps that I'll show for the phytoplankton species, we will have 4 alternative sets of predictors that alternatively include sea surface temperature at a monthly scale, the annual sea surface temperature, the only predictor at an annual scale, photosynthetically available irradiance, the log of chlorophyll A as a proxy for phytoplankton biomass, nitrate, silicates, N star and SE star. N star is the excess of nitrate relative to phosphate following the Redfield ratio, and SE star is the excess of silicate relative to nitrate. And for the zooplankton, well, we have also SST, annual range of SST, oxygen at depth, so it's not too collinear with oxygen at the surface, so we can capture oxygen minimum zones, hopefully. And yeah, the same micronutrients predictors. So in the end, we're able to model no less than 860 species, 336 for phytoplankton, 524 for zooplankton. For each species, we run 4 types of models, so generalized linear models and generalized additive models, which are multivariate regression models of various complexity, random forest, which is a classification tree, sorry, algorithm with machine learning, with boosting, and artificial neural networks. And we choose the set of parameters in each of these models to, again, avoid overfitting the observation and make spurious correlation projections. Each of the models sees 4 pools of different predictors, and as I said before, we perform several cross-evaluation runs. So at the end, per species, we have 160 models that are projected on the 12 monthly climatologies for the predictors they include. And so we do the sum of habitat suitability to estimate monthly species richness, and then we derive annual species richness by averaging them. And then, well, to estimate climate change, well, we just do the same, but into the future. So we derive, we use the output for 5 Earth system models, whose name I put on the left-hand side here. And for the predictors included in the statistical models, we derive the future monthly climatologies by using a delta approach, which is described here. And we just make the same predictions, but for the future conditions, and then look at the differences between the future minus the contemporary conditions. And I hope that's clear enough, but again, all the methods and very thorough descriptions are provided in the paper. So I'm just going to drink and show the main results of the study. First, the contemporary patterns. So the mean annual patterns of diversity for phytoplankton on the top, zooplankton in the middle, and all together at the bottom. And again, this is the sum of habitat suitability. So in yellow here, what we show are the regions of the ocean where the environment on the mean annual scale is favorable for most of the species. And so both trophic levels show an increasing diversity gradient from the poles to the tropic, which is quite standard for marine taxa. But there is a difference in the location of their peak in diversity. So for phytoplankton, what we see is an emerging diversity peak in the tropics, and particularly in the eastern boundary upwelling. We also see a peak in diversity in the tropics, but then more towards 30, 35 degrees north and south. As it gets actually too warm in the tropic, near the equator, the diversity of zooplankton tends to decrease, as not that many species seem to accommodate the very high temperatures. And this matches very, very well the previous observations that were made for zooplankton diversity pattern. We can also, of course, then disentangle that. I'm just showing you the results for higher trophic level, for very coarse classification, trophic levels, but of course we can then disentangle that into different PFTs and so on. So we can have a look at this for krill, for copepods, for diatoms alone, coccolithophores, jellyfish. And yeah, that's it. And what happens if you warm up the system? So into the future. So what I'm showing here is the difference in percentage relative to the contemporary prediction, to the contemporary conditions in species richness again. So basically on the maps, everything that's red corresponds to the potential increase in diversity. And in deep red are the regions where we expect an increase in diversity of more than 50%. And in blue and light blue will be the regions where we expect a decrease in diversity. And you can see that both trophic level show very different patterns. So for phytoplankton, we expect an increase in species richness of, on average, on median, 16% at the global scale, but with differences across latitudinal bands. So we expect a stronger increase in the tropical band, as phytoplankton, most of the phytoplankton species seem to accommodate the warming, and also a smaller increase, still an increase in the temperate and subpolar latitudes. And at the global scale, this is partially offset by a modest decrease that is expected north of 70 degrees. So in the Arctic, but I must say that in this region, we have much higher uncertainty due to disagreement between the model members. The members in the ensemble of models that we used. For zooplankton, you see that the spatial pattern is quite different. On a global scale, there is a we expect a relatively little change, with the median increase of plus 0.4%. However, this is low because this is the result of two contrasting patterns between regions. So in the temperate and subpolar latitudes, we project relatively strong increase by, on average, 24%. As you can see, in the North Atlantic and in the temperate latitudes of the Southern Hemisphere, we have very high increases in diversity. And this is partially offset by the slight decrease in the tropical band and in the Southern Ocean. So the two trophic levels show very contrasted patterns. And I can't go into the details. I'm speaking too much. Sorry, that always happens. But basically, the reason behind that is that both trophic levels do not accommodate to the same level. Their diversity does not respond in the same way when it comes to very high temperatures. So what we show here on this plot for phytoplankton and zooplankton is the log on the y-axis of species diversity as a function of mean annual thermal energy, which is derived from sea surface temperature. And what you can see is that actually, when you're past 22 degrees, phytoplankton diversity seems to keep increasing, seems to keep up with the warming. Whereas for zooplankton, there is an inflection point that is around 25 degrees, after which zooplankton diversity seems to decrease, which also explain at the beginning the differences in the positions for the diversity peaks. And in red, I've put the future tropical SST range that is derived for the models we use. And here is explained basically what is the main driving force behind the different patterns. And we can discuss later on why that is, but we're using statistical models which don't have processes directly coded into them. So we can, for now, only discuss what could be the potential cause behind that. So as you warm up the system, as you go, basically with global warming, we're going to push the system towards the right-hand side here. What's going to happen is that we're going to increasingly favor warm water species over cold water species. So what we wanted to ask then is from the projections, okay, we see increases in diversity in the high latitude, and we wanted to check whether that corresponded to shifts in community composition. So what we did is that for each species, we estimated the shift speed in kilometers per decade of the centroid of the global distribution. And we actually verified this hypothesis. So nearly 80% of all plankton taxa shifted polewards, 57 shifted westwards, which means that the other half shifted eastwards. So there's way more conflict into this direction, but maybe it's less interesting. 67 of the phytoplankton species migrated polewards, and 87, so way more, of the zooplankton species also migrated polewards. And the median shift speed between the two was not significantly different, but this is simply due to the fact that they're projected with the same type of models into the same outputs. And of course, dispersal limitation or dispersal capabilities were not included in these statistical models, so they come up at the end with the same shift speeds. So what does it mean in terms of community composition? Because if you only look at change in species richness, or the number of species, doesn't give you a complete overview of the system. Because if you have a difference in species richness of 0, it could also mean that you've lost 10 species, and that these 10 species were gained by 10 newcomers, which could have potentially could have huge implications for ecosystem function. So we wanted to quantify that, the turnover at the mean annual scale. So to compute that, we use the assemblages compositions in the present and in the future, and we use the Jacquard's dissimilarity index that can be decomposed into additive components, nestedness and turnover. So this is a composition dissimilarity index. The nestedness is basically the component that will tell you the dissimilarity that is driven by changes in richness, whereas the turnover is the component that we're interested in, and that is shown then mapped on the left-hand side, again for phytoplankton, zooplankton, and all. Both trophic levels seem to show actually similar turnover rates, with a global median value of 18%. It is slightly higher for zoo than phytoplankton, but again, there are strong regional differences, and particularly when you compare the tropics, where the turnover is relatively low, to the high latitudes, where there are several places, particularly the Arctic Ocean, where we expect a very sharp change in species composition, as well in the high latitudes that display a turnover of more than 40%, which is huge, and goes along this hypothesis of the boralization of the Arctic community with climate warming. Just to finish, maybe the last slide, we wanted to somehow try to understand what could be the impact of these changes for marine ecosystem services. So NPP, the flux of organic matter at depth, the efficiency of the biological carbon pump, and the quantity of fishes that are caught. But of course, with these models, we cannot build a strong link between a number of species and these ecosystem services. So what we did, we rather went for an overlap analysis where based on the changes in phytoplankton, zooplankton, their turnover, and the overall dissimilarity in the community, which we summarize with PCA, which is a dimensional these PCA scores, and we perform a very thorough hierarchical clustering approach to define the regions according to the strengths of their changes in richness and composition. And these are the regions that I show then here. And they're color-coded basically on the sensitivity of their plankton community to climate warming. And then, now based on the contemporary conditions of these regions, we look at how this region, what the distribution of proxies of ecosystem services for this region. So, formally speaking, normalized oceanic species richness, which is the number of fish species, marine mammal species, shark species, and so on. The quantity of POC, particulate organic carbon, exported below the euphotic zone, and PP, the ratio of the two, the E ratio, which is an indicator of the efficiency of biological carbon pump. The plankton size index retrieved from satellite observation. So the higher the index, the larger the particles and the plankton in these regions. And the catch rate of small pelagic fishes derived from the paper by Reg Watson in 2017. What you can see here, what came out from this overlap analysis is that the North Atlantic and the Arctic Oceans emerged as the most sensitive regions in terms of changes in species diversity and composition. And it happens that these regions, so in red, show relatively high rates of net primary production, but also they seem to be the community there seems to be quite efficient at letting the carbon go. So very efficient biological carbon pump with very relatively high particles and plankton cells, so larger animals. And these happen to be also one of the regions where people seem to fish the most. So potential strong impacts of climate change on those plankton-mediated marine ecosystem services. And then temperate latitudes follow somehow the same pattern, but they are a bit less sensitive to future changes. And meanwhile, yeah, tropical dryers and all the other regions were way less sensitive. And we have some regions like the Southern Ocean or the Peruvian upwelling, which are regions where the biological carbon pumps seem to be quite efficient, where net primary production is high, and where the future changes in plankton plankton richness and composition were not so high. So you're still, from this approach, that we still have places where it seems like the proxies of ecosystem services are quite high and the future changes not so strong. So a hopeful thought to forget. And I'm just summarizing the main results at this point. So I won't read all of that. I'll just leave it here maybe for discussions, but just wanted to let you know that then from this approach, we are now able to model the distribution at a monthly scale, global, for more than 860 taxa that are distributed across 10 different plankton functional types. So if anyone's interested, we're very much willing to share that. And yes, we have tons of research outlooks stemming from this research, and hopefully we'll present them in the future to JETZON. And with that, thank you a lot for listening. And if you want more details, please feel free to read the paper and go through the multiple appendices that we had to implement for this study. Yeah, with that, thank you.
Thank you very much, Fabio. Another talk with a huge variety of stuff within it. We're a little over the hour, but still got time for a few questions. If people have got some questions they would like to ask, let me just see if there's anything on the chat. Nothing on the chat yet. If anybody's got anything they would just like to ask over an open microphone, that's also fine. Maybe if I could just sort of get started in terms of you might have mentioned this and I might have missed this, but in terms of your projections and the changes in the diversity, can you separate out, for example, changes in the nutrient environment from changes of the physical environment as drivers? You know, for example, temperature versus iron availability.
So yeah, I actually did that a bit later. I could not include it in the study, but I always have it back in the back of my mind. I wanted to publish it somewhere else. So what you can do for that is when you perform your future projections, you keep one predictor, one parameter constant, and you only
Right.
Oh, you keep most of the parameter constant except one, and then you can evaluate how this parameter contributes to the overall change. So what I did is that I was specifically interested in quantifying the impact of SST, which is the main driver both in the models themselves, but also then in the future patterns. So if you keep oxygen, macronutrients, mixed layer, and so on constant, and you only change SST into the future, you end up explaining a lot of variance. So SST is the main driver. But what is very interesting is that depending on the functional group you look at, this changes. So if you look at cocolithophores, most of the variability into the future is not explained by SST. It seems like irradiance has a much higher impact. Another example, I don't have it I don't have it right now, but yeah, we did that. So to study the importance of the factors, you can do this. What we did first in the selection procedure is we look in the model. We do permutation tests, basically, where we also keep one, when we reshuffle basically one predictor, randomly, and we keep the others as is. And by comparing the output from this model to the unreshuffled model, then you have an idea of the importance. And do I have? Oh, I thought I had them in the appendices for this presentation. I should have prepared better. Clearly, SST turns out, especially for zooplankton, a bit less for phytoplankton. SST stands out as like the main predictor. And for phytoplankton, you have, interestingly, you have other predictors like N star or SE star or sometimes silicate that, depending on the functional group you look at, can overcome SST. Yeah.
Excellent. I mean, it sounds like there's another talk you can give us there, actually, get into that one. So I don't see anything else in terms of the chat, and because we're sort of over that time, I think we'll lose people. I think we will wrap up there. So thank you, Fabio, very much.
And feel free, I mean, if people have questions afterward, like they can contact me whenever they want. I'll be happy to answer anything. Thank you.
Excellent. Thank you very much. And otherwise, that's it for this month, and I hope to see many of you again for next month's call, and there will be a date going out for that in the very near future.